everyone, my name is Amy Wong. I'm a second year master's student in the Interdisciplinary Marine and Estuarine Sciences program at San Francisco State University. My project is using high throughput sequencing to determine the diets of these two copepods that you can see here on the left, Eurytemera caerulea and Pseudodactylus forbici. These two species are really important food sources for some threatened and endangered larval fish that live here in the estuary. So it sounds great, right? However, these copepods are not abundant year-round. They fluctuate in abundance throughout the seasons. For example, in the spring, Eurytemera declines while Pseudodactylus rises, and the opposite is true for the fall. The big question is, why does this happen? My hypothesis is that food shortage is driving transitions in copepod abundance. So to see if this was the case, I did some field work at one site in the spring and another in the fall to collect samples through the transition period, collecting copepods to see what are they eating and see what's inside of their diets, as well as water samples to see what the prey availability is within the environment. Next in the lab, I did two experiments, one to compare their diets to see what would they eat if given the same prey from the summer, and a starvation experiment to see what their gut microbiome would be like if they were starved and had no food at all. Then I did some molecular work. Currently, I am in the process of extracting all the DNA from the copepods and the water samples. And later down the line, I want to do high throughput sequencing. And using a method called metabarcoding, we're able to actually identify many different species from a single sample, which is really important to do work like diet composition. So as you can see from this graph, and these are just my expected results. These graphs are just examples and not from my data. But a diet composition would look like this, all the different colors representing the relative read abundance percentages of all the different taxa, whether it's inside of the copepod guts or within the environment. Next, I would want to make line graphs through each transition season to show the trends in copepod abundance as well as egg production rate. Lastly, I would want to do principal component analysis, which shows the similar groupings between samples. So thank you for hearing about my thesis research. I hope that you enjoyed it. And if you have any questions, please feel free to send me a message. Thank you.